Hi, my name is Arusha Shrivastava, and I'm currently a first-year undergraduate student at Caltech.
Hi, my name is Ariana Nair, and I am a rising junior at Rick Reedy High School in Frisco, Texas.
My name is Raymond Gow, and I'm currently a high school senior at Grafton High School in Yorktown, Virginia. And I'll be an incoming freshman at University of California, Berkeley in the upcoming fall.
So, Arusha, what is Trichomonas vaginalis?
Yeah, so Trichomonas vaginalis is a unicellular parasitic protozoan that is responsible for trichomoniasis. And this is the most prevalent non-viral sexually transmitted disease globally. It's also associated with the rising incidence of a variety of critical conditions like prostate cancer, HIV, HPV, and infertility.
So, Ariana, why was this study important?
This study is important because it brings awareness to the protozoan itself, whose molecular mechanisms are often not understood. Examples include its immune evasion through productions of homologs of host proteins, like MIF, also known as macrophage migration inhibitory factor, which the study brings a focus to.
So, Ariana, why did you want to participate in this project?
I initially wanted to participate in this project to gain an insight into structural science research and the process involved in studying various molecular structures. I previously did research focusing on socioeconomic disparities and inequities in terms of line act distribution, which drew me into cancer-focused research. Trichomonas was the perfect place to start in terms of this due to its widespread proliferation, opening my eyes to the significance associated with studying these structures.
Arusha, why did you want to participate in this project?
Yeah, so I want to participate in this project because of my strong interest in cancer research. Specifically, I wanted to learn more about the molecular disturbances that promote tumorigenesis. Previously, I worked on a lot of computational tool development for cancer analysis and fell in love with the topic. So I ended up joining Dr. Asoho's group to learn more about the biology behind cancer, and as a result, worked on this project with TvMIF and its association with prostate cancer.
Raymond, what were the most significant challenges and the best aspects of this virtual experience for you?
So the most difficult challenge for this project was definitely the time zone that everyone was in, since each of us had to schedule the same time for meeting and work on the project cohesively. And by being in a different time zone, it was really hard for us to make a specific time for everyone to meet and collaborate together. And the best aspect of this project was able to collaborate with leading scientists like Dr. Soho and students across the nation and gain hands-on experience with protein structure analysis, and then finally contribute to a publishing paper as a high school student.
So, Ariana, what was the most surprising or unexpected thing you learned during this project?
The most surprising thing that I learned was the highest incidence of trichomoniasis is found among incarcerated women. This highlights how infectious diseases often disproportionately affect marginalized communities, revealing deeper systemic issues tied to healthcare and social inequities.
Same question to you, Arusha.
Yeah, so I think the most surprising thing I learned from this project was about the huge topological similarity between human MIFs and TvMIF. And this is quite significant because it can be utilized for drug discovery, which exploits this structural congruency.
Okay. Same question to you, Raymond.
So the most unexpected and surprising thing that we learned in this project was definitely the use of PyMol itself, where we can use Python, which is the coding language, to construct the figure itself, or use the default buttons that's built within PyMol to construct different figures and analyze them.
Thank you for that. Now, the final question is, has this experience influenced what you want to study or do in the future? And I'll start with you, Arusha.
Yeah, so currently I'm an electrical engineering student with a minor in biology at Caltech. And through this project, I was able to further amplify my interest in biology. And in the future, as a result, I want to become a cancer biology researcher and also an entrepreneur that focuses on cancer technology.
Ariana, same question.
Yes, this has. As I was initially looking into medical anthropology as a profession, as I mentioned previously, that I was looking into research in that area. And this study expanded my horizons towards microbiology. I initially wanted to participate in this project to gain an insight into structural scientific research. And Trichomonas was a great place to start in terms of learning about structural microbiology because its complex ultrastructure offers insight into eukaryotic adaptation in anaerobic environments. The complexity of its molecular mechanisms, such as immune evasion, showed me the value of structural microbiology in understanding and addressing real world problems.
Raymond, same question. As this experience influenced what you want to do in the future?
Yes, this experience shows me how structural biology and computer science can work together to uncover the molecular drivers of diseases like cancer and uh, diseases. In the future, I hope to develop computational tools and technologies that can detect or distribute cancer-related proteins, leading to a more precise diagnostic and targeted therapy system.
Thank you so much. This was very nice talking to you about this project. Thank you.